one, B. Okay, right. All right. What I'm doing here is just working through some practice problems that I have set for you guys. So I'm going to start by opening up the okay, I'm going to start by opening up the sheet of practice problems that I have posted. Can everybody see this? It says Biology 1400 Genetics Practice Problems 1. Okay, hit me on the chat if you can't see this. If you don't say anything, I will assume that you are perfectly able to. I'm going to try to set that up for the best resolution I can, in case the screen is tiny. Hang on, view. Let's. Okay, I'm assuming you can all see this. The first problem I've given you is most corn plants have green leaves, but there's some that have a pattern of green and white stripes on the leaves, which for some reason is called IOJAP. I'm not sure why it's called that, except I suspect that the IO is short for Iowa, because the universities of Iowa, University of Iowa and Iowa State research on corn. Iowa is kind of all about the corn thing. And the first one is that a plant breeder crosses a green-leaved corn plant with an IOJAP corn plant, one with stripy leaves, and finds that the offspring all have green leaves. So which trait is dominant? Well, all of the offspring have green leaves. They don't have any kind of half and half state, not partly IOJAP and partly green. None of them look IOJAP, so we'd say that green leaves is dominant to IOJAP. So that one should be fairly easy. It's the same kind of reasoning that we worked through with pea plants with purple flower color being dominant to white flower color. Okay, and B, the breeder takes one of the hybrids from question 1A above. That's one of the green-leaved offspring that he got up here, and crossbreeds it with an IOJAP corn plant. So let's predict the results of the cross using a Punnett square. Okay, let me see how this Jamboard thing works. Okay, that seems to be my jam. Can everybody see this? Should be a kind of white area, and I can, doodle on it and, okay, draw stuff. All right, I will assume that everybody can. So we want to do a Punnett square, and we're crossing a green hybrid corn plant with an IOJAP plant. Now, the F1 offspring that we're starting with is green-leaved, but we know that one of its parents was IOJAP, so its genotype has to be, I'm going to write big G, little g, because it's got to be heterozygous, and I'm going to use big G for green and little g for IOJAP. There, make that look a little bit more like a G. And the other plant that we're crossing, I'm going to draw a cross, is IOJAP itself, and that's recessive. So for it to look IOJAP, it's got to have 2 copies of the little g recessive allele. Okay, so we start by drawing our Punnett square. Okay, that sort of looks like a square. This parent right here has green leaves, but it's heterozygous. This one can pass on one of each pair of alleles, so it will either pass on its dominant allele, which we represent with big G, or its recessive allele, can't pass on both. So I can write here either a capital G or a lowercase g. What we write up here at the top are all of the possible alleles or allele combinations that a given parent can pass on. So this one can pass on either that big G, or it can pass on its little g, not both. This one over here, the parent that is IOJAP, can only pass on a little g allele. This one can either pass on the first little g allele. Okay, I swear that's a G. And that's sort of a G. It can pass on either the first little g or the second little g, but that's redundant. There's no difference between them. So we've set up our 2x2 Punnett square, and we fill it in by moving the letters down the rows and left to right across the columns. So in this box, we put big G, little g, and little g. All right, so we've taken those letters and filled them in on the rows from top to bottom. And from left to right, we fill in little g, and then we move this from left to right, little g, that's a G, and little g. Okay, so there's our Punnett square. What I've asked you for is the genotypes and phenotypes. Remember that genotype refers to a combination of alleles. So half of our offspring, 2 out of 4, which is the same as 1/2, are going to have the genotype big G, little g. They'll be heterozygous, and the other half will have the genotype little g. They will be homozygous. Okay. As for the phenotypes, remember, phenotype is a description of a trait or a feature. In this case, the phenotypes would be green leaves or IOJAP leaves. Green leaves or IOJAP is, green and white stripy. So in this case, half of the offspring would have green leaves. They will be the ones that are heterozygous. And half of the offspring will have IOJAP leaves. Those are the ones that will be homozygous. So half of our offspring will have green leaves and half will have stripy leaves. Okay, I'm going to cut over here and check the... Okay, Stephen says yes, and green. Okay, you've got it. All right, let me cut over for a second to the next practice problem. Which looks like this. Here we've got to do, okay, that first part was really easy, but in 1C, what we have to do is a cross that involves looking at 2 genes, each with 2 alleles. And it turns out that a corn gene called sugary has 2 alleles. One allele, the kernels are starchy. Corn like that you'd probably want for making cornmeal or something like that. And the allele for starchy kernels is dominant to the allele for sugar-rich sweet kernels, which is what you'd probably want if you're growing corn for eating fresh. If you're eating sweet corn, people tend to like really sweet kernels. So our plant breeder crosses a homozygous starchy green-leafed corn plant with a sugary iojap corn plant, and all of those offspring have green leaves and starchy kernels. Now she crosses one of those F1s with another plant that is sugary and iojap. So let's cut over to the Jamboard. Okay, there's where we are. There's a second page. So our initial cross was we were taking a plant that was homozygous for green leaves and starchy kernels. We can write that as big G, and I'll use capital S for starchy. And we cross that plant with a plant that is iojap and sweet. So that plant will have a genotype that looks like little g, little s, little s. Notice, by the way, I'm writing my lowercase s's in cursive. You don't have to do this. I mean, it's going to be, pretty much moot as far as grading goes because you're going to be typing in your answers, I assume. Certainly you will on the test, but it's still important not to confuse yourself. And if you're doing this in handwriting, it's easy to mix up a capital and a lowercase s if you block print them both, especially if you're working on paper that hasn't got lines. So I recommend that you do that just to keep from confusing yourself. And the offspring that you get, remember that the offspring are all going to get one of each pair of alleles from each parent. So we could do a 1-by-1 Punnett square, but we don't really need to. Think about this. This parent right here will contribute one of its alleles for leaf color, which in this case has to be big G, and one of its alleles for kernel taste, which in this case will be starchy. This parent is going to pass on one of its alleles for leaf color, which in this case will always be iojap, and one of its alleles for kernel flavor, which in this case will be sweet. So all of the offspring are going to get one green from this parent and one iojap from this parent, and they'll all get one starchy from this parent and one sweet from that parent, and they will be heterozygous for both genes. Their phenotype will be green leaves and starchy kernels. Their genotype will be heterozygous for both, big G, big S, little s. What you have to work now, the problem is that we take one of these and we're going to crossbreed it with another plant that is iojap and sweet. So we're going to cross this with another corn plant whose genotype looks like this. So how do we do that? Here's where we're going to need our Punnett square. Here's our square. Okay, close enough. Now think about this parent right here. That one, there are 4 possible combinations of alleles that this parent can pass on. It can pass on the first of each pair, with capital G, capital S. So we'll put that up here. Or it can pass on the outers of each pair. So that's capital B and lowercase s. can pass on the inner of each pair. So that's lowercase g and capital S. Or it could pass on the last of each pair, and that's lowercase g and lowercase s. Okay, those are the four possible combination alleles that this parent can pass on in its either egg cells or sperm nuclei, whichever is the case. In some cases, that will matter. In this case, it does not. So you take the first and the outers and the inners and the last, and we have the FOIL method, which I'm sure you remember from high school algebra. We can do the same thing for this parent. We can take the first of each pair, the outers of each pair, and we could take the inners of each pair, and we could take the last of each pair. But in all cases, all we're going to have is little g, little s. Right? So if I really wanted to, I mean, really, I could work this with only one row and make it a Punnett rectangle. But I'll go ahead and fill in the columns just because, you know, I'm here. All right, this is kind of laggy, meaning I can't write as fast as I'd like to, but so it goes. And now we can draw our rows and columns. Like so. So we've got a 4x4 Punnett square. Okay, what do we put in our rows? Well, we just take the letters from the top, or what do we put on our columns? We take the letters from the top and move them down. So in this box, I'll put big G, big S. And I could write them in, you know, these other columns, but they're all going to be the same. So I'm just going to put little, you know, quotation marks. Where? And in here, I'm going to put big G, little s. And here I'm going to put little g, big S. And here I'm going to put little g, little s. Okay, there's the columns. How about the rows? Well, I'll take these letters over here at the left-hand side of the column and fill those in going left to right. So here is little g, little s, and little g, little s, and little g, little s, and little g, little s. Okay, I'm not going to fill in the other blocks because, again, the lower three rows are all going to be identical to the top row. I guess I don't even really need to put in the quotation marks. This won't always be the case. There will be some Punnett squares where you have to fill in all 16 squares, but this is one where we can, you know, take a shortcut just a bit. And if we look at what's in here, this box right here, big G, big S, little s, this one is going to be heterozygous for both genes. And the phenotype is going to be green leaves and starchy kernels. So this one's green and starchy. This one's going to be green but have sweet kernels, green and sweet. This one is going to have IOJAP leaves, green and white striped, but have starchy kernels. And this one represents offspring that will have stripy leaves and sweet kernels. And the prediction is that we should get equal amounts of all four. So our prediction is 1/4 of our cross, 1/4 of the offspring from this breeding experiment that we've just done will have green leaves and starchy kernels. 1/4 will have green leaves and sweet kernels. 1/4 will have IOJAP leaves and starchy kernels. And 1/4 will have IOJAP leaves and sweet kernels. 1/4 will have the genotype big G, little g, big S, little s. 1/4 will be big G, little s, little s. 1/4 will be little g, big S, little s. And 1/4 will be little g, little s, little s. Okay. All right. I'm going to come back here to the meat for a second. And I'm going to go ahead and check. Nobody has posted anything new on the chat. Oh dear, I am seeing that somebody got booted off, or maybe left on her own accord. I'm sorry about that glitch. That just seems to happen. Would you like me to work one more problem? Because I totally can. Okay, what the heck? I'll do it anyway. So let's get back over to the practice problem set. And let's see, 2 is fairly easy. Let's do 3. Okay, 5 is kind of fun. Okay, in 5, a breeder of hamsters crosses a golden hamster with a black hamster, and all the babies are dominant. Sorry, all the babies are golden. Which trait is dominant and which is recessive? By now you should see that gold is dominant to black. What are the genotypes of the parents and the genotypes of the babies? Well, the parents must be homozygous dominant and homozygous recessive. So that would be, we could write that as big G, little g, or sorry, big G, and little g, and the babies would all be big G, little g. We take, actually 5 is, believe it or not, that's the same problem as 1. Okay. Oh, yeah, I actually did remember to include the, okay, that's a slightly more interesting one down there. I just realized that 5 is basically the same problem as 1. So let's do 3. This one's about turkeys. In turkeys, bronze body color is dominant over red. Normal feathers are dominant over hairy feathers. If you made a bronze male turkey to a bronze female, some of the babies produced by this cross are red. What are the genotypes of the male and female parents? Well, let's see. Let's cut over to the board. Okay, there's our clean sheet. Okay, so we mate a bronze male turkey to a bronze female turkey, and some of the babies are red. Well, we already know that bronze is dominant over red. So if we're crossing 2 bronze turkeys, for them to be bronze, they have to have at least 1 copy of the dominant allele, which we can write as big B. Okay, we know that some of the offspring are red. So we don't know how many are red, but we actually don't need to know. some of those offspring have this genotype. Okay, so we're crossing these two parents. We know they're bronze, so they've got to have one copy of the big B allele. We don't know what the other allele is, but we do know that some of the babies are little b, little b. They have to be because they're red. Some of the babies are red. The only way for them to be red is to have two copies of that recessive red allele. The only way they can get that is that if they get one red allele from mom and one red allele from dad. That means that each of the parents must have a red allele right? We know that they must have one bronze allele because they look bronze, but if they have any offspring that are red, the only way you can have red offspring is for the red offspring to get one of the recessive red alleles from mom and one of the recessive red alleles from dad. So both parents must have that genotype. So that's it. We've worked out the genotypes of the male and female parents and the red poults. That's baby turkeys. Okay, some of the poults from the cross described above are bronze. What proportion of the poults would you expect to have the different genotypes? That we just solved by doing a good old-fashioned Punnett square. We have this parent, and here's our square. Or, semi-regular trapezium or whatever it is. And this parent can pass on either his or hers. In this case, it doesn't matter which parent is which. Tomorrow we'll see a case where it does, but right now let's not worry about that. So this parent up here can pass on either his big B allele or his little b allele, but he can't pass on both. This parent can pass on either her big B allele or her little b allele, but not both. So that's what we write. And we just fill in the letters going down and across. So we put a big B here and a big B here and a little b here and a little b here. And then we take the letters on the left and move them down rows to the right. So there's a big B here and a little b here and a big B here and a little b here. And sorry, I just heard a ding dong. All right, good morning. We're working our way, since you just came in late, through a set of practice problems that I've just posted on the virtual classroom, and I'll show you what they look like in just a minute once I finish this Punnett square. Over what just happened? Oh, okay, she's still here. Right. Okay, so I filled this in. I wrote little b, big B over here. That's not necessarily wrong, but the convention is that we usually put the dominant alleles before the recessive alleles. So it would be better style to do it this way. Although in the past I haven't necessarily marked that off. It doesn't really matter what order you write the alleles in, but it usually just seems more of, the accepted standard to put the dominant ones first. So there's our completed Punnett square. 1/4 of our turkey poults, that's baby turkeys, will have genotype big B, and they will look bronze. Half of our turkey poults will have genotype big B, and they will look bronze. And 1/4 will have the genotype little b, and they will look red. So that takes care of that much. So I'm going to cut back to the practice problems. And we'll do the last one, and then I'm going to, as my wife's family says, bless this and call it dinner. So we want to cross a big B, little n turkey with a little b, big N, little n turkey. And if you forgot, N is for normal feathers, and little big N, normal feathers, is dominant over little n, hairy feathers. So we want to cross a big B, little n turkey with a little b, big N, little n turkey. What does that even look like? Well, let's get over to the board. And I hope bored is not what you are, at least not too badly. All right, let's get over to another page. And we're crossing one turkey that has the genotype big B, little n, little n. Think about that. That's a turkey that's going to be bronze, because bronze color is dominant to red color. And it's going to have hairy feathers, because hairy is recessive to normal. And we're crossing that with a turkey with the genotype little b, big N, little n. So that's going to be a turkey with red feathers, but normal texture. They'll be normal feathers, not hairy feathers. And the question is basically asking us to predict the results of this cross. So we put our turkeys together and see what they do. And if they are the most common breed of meat turkeys, which are called broad-breasted white, they do absolutely nothing because they're colossally stupid and you have to inseminate them artificially. But actually, traditional domestic breeds are still reasonably smart. So these turkeys are probably intelligent enough to figure out how to mingle their eggs and sperm. This parent right here, if you want to, you can do the whole first, outer, inner, last. There's really only 2 possible allele combinations that this parent can pass on. This parent can pass on, this parent will pass on one of its B's and one of its N's. And so this parent can pass on either big B and little n. That looks like a 13. It's a B. Or the other possible option is it can pass on a little b and a little n. This parent, you can figure this out with the FOIL method if you want to, but this one really can only pass on 2 combinations, a little b and a big N, or a little b and a little n. So we really only have a 2 by 2 Punnett square. All right, so we move the letters down and across. In here, I'm going to put big B from this parent, little b from this parent, big N from this parent, and little n from that parent. Okay, and as I'm moving these letters down and across, I'm sorting them so that I group like letters with like letters, and so that I'm putting the dominant alleles first. I mean, you could write little b, little n, big B, big N, or something like that, and it would still technically be right, but it's much easier to follow if you just follow the conventions. So in here, I'm going to put big B, little b, little n, little n. And here I'm going to put little b, big N, little n. And here I'm going to put little b, Hey, hi, sweetie. Are you busy? I'm almost done. Okay. What's up? Oh, you're not on camera? I am. Oh, cool. Okay. Yeah. Okay. Okay. When I said sweetie just now, I meant my wife has just woken up. I'm sure you're all sweeties to somebody. You're not specifically my sweeties, although I'm sure you're all nice people. Right. Anyway, so this is the answer we were looking for. 1/4 of our offspring are going to have the genotype big B, big N, little n, and they're going to look bronze with normal feathers. 1/4 of our offspring will have this genotype, big B, little n, and their phenotype is they'll have bronze, hairy feathers. 1/4 of our offspring will have this genotype, little b, big N, little n, and they will have red, normal feathers. And 1/4 of our offspring will have that genotype, little b, little n, and they will have red, hairy feathers. So 1/4 of our offspring will be heterozygous for both, 1/4 will be homozygous for both, and the other 2 will be homozygous for one gene, but not the other gene. So that is the answer we were looking for. I'm going to go ahead and cut back here. If anybody's got questions, go ahead and put them in the chat if you want to. I'm going to go ahead and stop recording, but I'll still be available if this has been anything less than clear. So here we go. I'll stop recording, but I will take questions